Good afternoon, everyone. So this is the first of our pre-recorded lessons and we're trying something new. So today we are going to be talking about genetic disorders and pedigrees and how to read a pedigree, how to work through it, how to understand what the pedigree is telling us about our families. So let's get right into it. I'm going to go ahead and share my screen with you. All right, so genetic disorders and pedigrees. Basically, we're going to be talking about how we can predict offspring ratios based on different inheritance patterns and a different variety of ways to read those inheritance patterns. So we talked about Punnett squares, where we calculated the probability of different genotypes and outcomes based on the parents' genotypes. But we're also going to be looking at pedigrees, which are a picture of family inheritances of a certain genetic disorder. So let's hop right into it. So your essential questions for this lesson are: how are pedigrees used to track inheritance of genetic disorders through a family tree? And also we're going to be constructing a pedigree based off of a case study. So you're going to be able to build a pedigree based off of a word problem given to you. So instead of just being able to interpret one, seeing one, you're going to be able to build one as well. So what is a pedigree? What is that? You know, we heard about pedigree dog food, but what are pedigrees for a dog, right? We're talking about the dog's parents, but a pedigree is just a chart that shows a trait and how it's passed from the generation to the next. So just like dogs have pedigrees that show their parents and what they pass on, we have pedigrees that show our parents' traits and what they pass on to us. So when you're looking at a pedigree, not only are we able to see how one trait is passed to one generation, from one generation to the next, we're also able to assess the genotypes and phenotypes of specific family members, so we can determine those specific genotypes and phenotypes. Remember, genotypes are your letters, 2 letters, a dominant allele and a recessive allele, or 2 recessive alleles, or 2 dominant alleles, right? Homozygous dominant, heterozygous, and homozygous recessive. And we can also see our phenotypes. So phenotypes are our physical traits. What do those genotypes actually show physically, right? So what can a pedigree actually tell us? When it's telling us how a trait is passed on, it's telling us whether this trait or disorder is autosomal or sex-linked, and whether it's dominant or recessive. Okay. So the first thing to understand a pedigree that we need to know, or we need to understand the symbols in a pedigree. Okay, so circles are females and squares are males. That is the first thing we need to understand. Circles are females and squares are males. Think about females, we're round, where our womb is soft and round, right? Our bellies get round when we get pregnant. Whereas a male, he's like a square, he's rough around the edges, he's really rigid. Well, that's the way I like to think about it. Not all males are rigid, but. So when it is not shaded in, an unshaded shape means that that person, whether a male or a female, is healthy. So an unshaded circle is a healthy female, because circles are females, and unshaded means that they don't have the trait. They're not filled with that trait. It's just normal. And an unshaded square is a healthy male, because squares are males, and unshaded means that they're healthy, they're unaffected. Okay. Here you have our half shaded in halfway, our shapes shaded in halfway. That means that they're carriers. So a carrier, if you hop over to my asterisk over to the side, carriers mean that the individual has a heterozygous genotype, meaning that they carry the gene for the trait, but they are not affected by it. So they carry that gene, but that gene is not affecting them. It's not hurting them or helping them, depends on what the trait is. Okay, so if it's shaded in halfway and it's a circle, it's a carrier female. But if it's shaded in halfway and it's a square, it's a carrier male. Now, beware, not all pedigrees use this shaded in circle or shaded in square. Not all pedigrees show carriers as shaded in halfway, so we just have to be careful about that. Affected females or affected males are denoted by shaded in shapes. So an affected female is a shaded in circle because circles are female. And if it's shaded in, that means she's affected. She has it. She's full of that gene. Okay? Whereas an affected male is a shaded in square because squares are male and it's shaded in. He's full of that gene. All right. If you see a line between a square and a circle, that means that they're married or they're mates. That means that they're mates. They're married. They're together. They were together at some point. Okay. If you see a line coming down from the middle and branching off into other shapes, that means that these are offspring of the parents. And if they're branched off, that these are siblings. So you have a mom and a dad. They're parents of a girl and a boy. Okay, so if you've ever seen a line branched off and it's connected, that means they are parents to their offspring. Okay. If you ever see a line coming from a single origin and it's a male and a female, they are fraternal twins. But if you see a line bridging from a single point and they are connected and the sexes are the same, they are identical twins, or it could be a male and female, but if they're connected, that means they're identical twins. But the main thing I want you to remember is that your parents of offspring have that branched line in the middle. And they section off to show their offspring. And if they're connected by this branched line, that means they're siblings. All right, so the second thing we have to make sure we understand is whether the trait is autosomal or sex-linked. So if you remember us talking about sex-linked traits in our Punnett squares, if you have not, please go back and watch that video under inheritance patterns. But an autosomal trait is caused by genes on chromosomes 1 through 22. So remember, we have 23 chromosomes we get from mom and 23 chromosomes we get from dad. 1 through 22 are your autosomal chromosomes. Remember, they control everything about your body. Your ears, your eyes, your nose, your throat, your skin color, your hair color. Those are non-sex chromosomes that just carry out normal functions and control all normal functions of your body. So if a pedigree, or if the trait is an autosomal trait, it appears equally in each sex. So each sex will equally have equal males and females will be shaded in. So if there are 3 males shaded in, then there are 3 females shaded in. Okay. Whereas if the trait is sex-linked, that means it's caused by, the trait or disorder is caused by genes on sex chromosomes, which are the X and Y chromosomes. So that 23rd chromosome that determines your sex, if the trait is on one of those chromosomes, either X or Y, it is determined to be a sex-linked trait. And on a pedigree, to tell whether a pedigree, a trait is sex-linked, is inherited as sex-linked, more males in the pedigree have it. So there will be more shaded in males than females. So let's look at this pedigree. Do you think that this pedigree is autosomal or sex-linked? Is there an equal amount of people in this pedigree affected, or are there more males than females? Yes, there is an equal amount of males and females affected. So this is an autosomal disorder. Most likely, this trait is caused by a gene on chromosomes 1 through 22. I'm going to stop right here for a second because we also need to understand that on a pedigree, there are such things, there are generations shown on a pedigree. Generations are shown horizontally. So this is a generation. This is generation 1. This is generation 2. And this is generation 3. This means that this first generation is most likely grandparents. So this is a grandpa and this is a grandma. This may be a grandpa and grandma, and this may be a grandpa. And as we go down the generations, who does grandpa and grandma give birth to? The parents. So this is a mom and this is a dad. And then these parents give birth to who? Their offspring, 2 boys and a girl. Because remember, males are squares and females are circles. Okay? So this is generation 1, generation 2, generation 3. The topmost generation is your oldest generation. These are your oldest human beings on your pedigree. Whereas the last generation of your pedigree is always the youngest. Oldest to youngest. Okay? So really quickly, if I asked you, how many siblings were a part of the 3rd generation, how many would you say? Right, 3. 1, 2, 3. Because these are siblings under their parents. How did we know that for their parents? Because this line right here shows them as being mates, connected. So the last thing we need to determine, the last thing we need to know to determine whether a trait or how a trait is inherited through a pedigree is determining whether the trait is dominant or recessive. So if the trait is dominant, one or both parents will have the disorder if the offspring does. And dominantly inherited traits never skip a generation. So dominant traits means that one or both parents will have the disorder if the offspring does. If one parent has it, then the offspring will have it because it's dominant. It will always show up. It's dominant. It will always show up. And since it's dominant and will always show up, if one or both parents has it, it'll never skip a generation because it'll always get passed on. However, if it's recessive, that means that neither parent has the disorder, but the parents can be carriers, which means they can be heterozygous. They can carry that recessive gene, but it won't show up unless both recessive alleles are passed on. And since recessive alleles only show up if both recessive alleles are passed on, it can skip generations because recessive doesn't always show up. So if the trait is dominant, it will always show up and never skip a generation. But if it's recessive, it can skip generations because it might not always show up. Okay? So let's determine whether this trait on this pedigree, whatever trait we're talking about, is dominant or recessive. So, does this trait show up in every generation? Or does it skip a generation? This is generation 1, generation 2, and generation 3. So, did it show up in every generation or did it skip a generation or two? That's right. It's a recessive disorder because the parents in the first generation got skipped. The parents in the first generation did not have it. But if offspring in the next generation have it or had it, what can you determine? That maybe these parents were heterozygous for the trait. And somehow they passed on that recessive allele to one of their daughters. All right. So let's talk about some genetic disorders and talk about their inherited, or talk about how they're inherited. So Huntington's disease is a common disease that degenerates or breaks down the nervous system. What this means is that the nerve cells in your brain break down slowly over time. So it causes you to move uncontrollably, you lose mental capacity, and you may even die early. Huntington's disease is an autosomal dominant disorder. So look at this, a healthy brain, look at how much light gray area we have. That means that's active, living brain cells, nerve cells that are sending impulses and signals. But look over here, in Huntington's, the brain tissue is decreased, significantly decreased. So let's look at this pedigree. Below is a pedigree for a family with Huntington's disease. Okay, so if it's unshaded, if the shape is unshaded, if the person is unshaded, that means that they're normal or unaffected. But if they're shaded, that means they have Huntington's disease or they are affected. Okay, so let's understand our symbols first. We have 3 steps. Understand our symbols, understand whether it's autosomal or sex-linked, and understand whether it's dominant or recessive. So let's look at this family with Huntington's disease. How is this trait inherited? Well, the first thing we have to see is whether it's autosomal or sex-linked. Remember, autosomal traits affect both males and females equally, whereas sex-linked traits, more males are affected than females. So, looking at this pedigree, Are more males affected than females, or are males and females affected equally? Let's count. How many males are affected? 1, 2, 3. 1, 2, 3. Okay. How many females are affected? 1, 2, 3. So it's 3 males to 3 females. They're equally affected, which means that this is an autosomal trait. Or it's an autosomal trait. Okay. The second step we have to figure out is whether the trait is dominant or recessive. So remember, if it's dominant, it never skips a generation. If it's recessive, it can skip a generation. So, do we have at least one affected person in each generation, or did it skip a generation? Right, we have at least one affected person in each generation. Remember, generations are denoted by each line. This is a generation, generation 1. This is a generation, generation 2. And this is a generation, generation 3. And it did not skip a generation because at least one person per generation is affected. So that means it's a dominant trait. So how is this trait inherited? Autosomal dominant. It's autosomal dominantly inherited. Excuse me. Okay. So the next thing that we've got to do is determine some of the genotypes. So how do we do that? For this, we can just pick a letter. Let's pick the letter H. And if we pick the letter H, then we can determine what traits are coded for by these alleles. So it says it's an autosomal dominant. So if it's dominant, that means the capital H must code for what? Huntington's. Because remember, capital letters in genotypes denote dominant alleles for the trait. So hunting, I'm just going to put hunting. So that means if you have 2 lowercase h's or a lowercase h, that means you're normal. Okay. So let's figure out some of the genotypes. How do we read this? 2, 3. This means that we are looking at generation 2. So here we go, generation 2, individual 3. Here's individual 3. So generation 2, individual 3, look at that. He's an affected male. He's an affected individual. So what are his 2 possible genotypes? We know he has Huntington, so he must have a capital H. So he could either be homozygous dominant, 2 capital H's, or he could be heterozygous, just as long as his genotype has 1 H, 1 capital H. Okay. Let's look at generation 3. Here we go. Generation 3, individual 1. Here's individual 1. She's not affected. So what must be the only possibility? She's normal. She's unaffected. She's unshaded. So what must be her only possible genotype? Good. Lowercase h, lowercase h. Because if she had one capital H, she would be shaded in, but she doesn't. She doesn't have this dominant trait. So she has two recessive alleles. She is homozygous recessive. Okay, let's see if we can figure out the phenotype. What is the phenotype of individual of generation 1? Here we go, generation 1, individual 2, individual 2. So she, remember she's a circle, so she's a she. Women are circles. She is shaded in. So that means if she's shaded, she's affected. So if she's shaded, what is her phenotype? What does she have? Yes, she has Huntington's. So her phenotype is that she has Huntington's. Okay. Oh, let's keep it pushing. Okay. Here's another example of an autosomal dominant disorder, polydactyly. Poly means what? What does poly mean? Poly means many. And dactyl means fingers. So polydactyly means that you have extra fingers, and it's a dominant trait. Wait, so you're telling me it's a dominant trait to have extra fingers? That means that we're all supposed to have extra fingers. We're supposed to have 6 fingers. However, we all have the recessive trait to only have 5. Pretty cool stuff. Side note, if you ever see like a little patch of skin, on the side of your pinky of your hand or on the front, that was almost a finger. Okay, so here are some examples of some autosomal recessive disorders, right? So sickle cell anemia. We had wonderful presentations on sickle cell anemia 2 weeks ago, right? So sickle cell anemia is when your red blood cells deform or they're not regularly shaped and they fold into this crescent shape. They look sickled and I can't do my job, right? And so this shape, they're not a perfect bowl shape. They can't carry oxygen or move through the vessels easily. So they end up clogging up the vessels and causing blockages. And that stops your blood flow. And do you think we can afford to have obstructed blood flow? No. So people with sickle cell anemia can experience weakness and pain, and it also can damage your brain and your heart and your spleen because your blood is your life supply. It carries oxygen to your tissues wherever you need it. So sickle cell shows incomplete dominance. Remember what incomplete dominance was? Incomplete dominance causes a mix. Okay? And since it's an autosomal recessive disorder, it's caused by a recessive allele. Remember, recessive alleles are denoted by lowercase letters. So someone who is homozygous dominant, 2 capital letters, homo, same, dominant, capital letters, is a healthy individual. So they have no sickle-shaped cells. So they have just regular bowl-shaped red blood cells, carrying oxygen, doing our job. People who are heterozygous right? They have two different letters, a capital A and a lowercase a, carry the sickle cell trait and have some sickle-shaped cells, okay? It's incomplete dominant. So there's a mix of healthy cells and sickle cells, right? So they carry that trait and they have some sickle cells. But people who are homozygous recessive, two lowercase a's, have sickle cell anemia and have mostly sickle-shaped cells, okay? They're diseased. But look at this. Sickle cell, even though it sucks, may not all be bad. So sickle-shaped cells are resistant to malaria. Okay, so malaria is a disease that's carried by mosquitoes in tropical areas, specifically and mainly in Africa. Okay, but people who have sickle cell are resistant to malaria because the disease piggybacks off of the healthy red blood cells. But since people with sickle cell don't have healthy red blood cells, that malaria cannot survive. So, though they may have sickle cell, they are resistant to malaria. Okay? People who are heterozygous, who have those sickled cells but also have healthy cells too, are genuinely healthy and malaria resistant. However, people who are normally healthy with no sickled cells can get malaria because that malaria can thrive in those healthy cells. Okay. Another example of a genetic disorder, specifically an autosomal recessive disorder, is called cystic fibrosis. Cystic fibrosis affects your glands that secrete sweat and mucus. Okay, so people with CF have thicker mucus in their lungs, which makes breathing really hard for them and also makes them susceptible to bacterial infections. So have you ever had a cold and you cough up that mucus and you cough up that green icky stuff? People who have cystic fibrosis, that's a lot thicker, especially in their lungs, and they can't breathe. And they get sick easier because that mucus is harboring that nasty bacteria and holding on to that nasty bacteria. So people with cystic fibrosis actually have to sit in the hospital for extended periods of time and get their chest beat for hours on end. Do you know how irritating that would be? Do you know how irritating that would be? Okay, can you stop? Nope, we gotta break up all the mucus. I'm done. It's only been an hour. And they just had to keep getting beat and beat and beat. It's like trying to burp a baby for 3 hours. It sucks. So, another kind of genetic disorder is called hemophilia, and it is sex-linked recessive, which means that it's X-linked. Hemophilia is a blood clotting disorder, which basically means that people with hemophilia don't have the ability to clot their blood. They're missing that protein that helps them to clot that blood. They bruise really easily and they can die of minor cuts. Since it's sex-linked, it's more common in males. Remember, sex-linked traits affects males more than females. Okay? So if you cut your arm shaving and you had hemophilia, you would just bleed and bleed and bleed let's look at this pedigree. Below is a pedigree of a family with hemophilia. Okay, so if they're unshaded, that means they're normal. They're affected. They're unaffected. But if they're shaded, that means they have hemophilia or they're affected. Okay? And our 3 steps to understanding a pedigree: you have to understand your symbols, you have to determine whether the trait is autosomal or sex-linked, and you have to determine whether the trait is dominant or recessive. So let's look at this pedigree. First of all, how many generations do we have? That's right, 4. 1, 2, 3, 4. Okay. Now, is this trait autosomal or sex-linked? Are males and females equally affected, or are more males affected? Remember, females are circles and males are squares. Let's count how many affected males we have. 1, 2, 3. Let's count how many affected females we have. 0. So it was 3 to 0. So this trait is sex-linked because more males are affected than females. Is this trait dominant or recessive? Did it show up in every generation? Did it never skip a generation? Is it dominant? Or did it skip a generation? Is it recessive? That's right. It's recessive because it skipped the first generation. So this is a sex-linked recessively inherited trait. Okay. So now let's determine the genotypes of these 2 individuals. So generation 2, individual 5. Generation 2, individual 5. Here we go. Since we're talking about sex-linked traits, please make sure you go back and review, if you have not, sex-linked Punnett squares in our inheritance patterns, because that will help you figure out genotypes when we're talking about sex-linked traits. So is this a male or a female? Is individual 5 a male or a female? Right, she's a female. So we know that she must be X. And for the sake of this, we can use what letter should we use? We can use, let's just use, let's use A, right? Let's use A. And if it's recessive, then we know, and this is a recessive trait, then we know A codes for hemo. And if there's a capital dominant letter, that means they're normal. Sorry, I didn't do that before, everyone. Okay, so let's look at the genotype for generation 2, individual 5. So she's a female, she's XX, but she has traits on these sex chromosomes. So she's not affected. So we know that she has at least one capital A. we know that she has at least one capital A because she's not affected. But this is where we look. This is where we look at her kids to see what allele might be on her second X. One of her kids, look at this, is affected. So she must be a carrier. So it would be a lowercase a. So remember I said in the beginning that not all pedigrees showed carriers as shaded half in. Ugh, and I can't even do it now. But she would be a carrier. How do we know she was a carrier? Because she's not affected. She's not affected, but her child is affected, which means she had the ability to give one recessive allele to her child. Okay. Let's look at generation 4, individual 1. So generation 4, individual 1. Since this is a sex-linked problem, a sex-linked pedigree, we have to write out our sexes. So it's a square. Is that a male or a female? Yes, it's a male. So it would be X, Y. It's my X. And remember, no traits are carried on the Y chromosome. It's too small. It only determines that a man is a man. That's it. No traits are carried on the Y chromosome. So X is going to carry this trait. What allele are we putting on our X? Is he affected? Is he shaded in? Or is he unaffected? Is he unshaded? He's shaded in, so he's affected. And this is a sex-linked recessive disorder, so he must have the recessive allele, which is a lowercase a. Okay. Let's determine the phenotype of individual 2 from generation 3. So generation 3, let me get an eraser. Generation 3, individual 2, generation 3, individual 2. What is the phenotype of this individual? It's a circle, so is it a male or a female? It's a female. And is it shaded in or unshaded? It's unshaded, but look at this. Her child is affected. So she must be a what? A carrier. So she is a unaffected carrier, female. That is her phenotype. She is an unaffected carrier female. All right, another sex-linked recessive disorder is called Duchenne Muscular Dystrophy. This is also X-linked, which means that it affects more males than females. And it's caused by a recessive allele, a lowercase letter. A person with Duchenne muscular dystrophy over time has breakdown of their muscles. and they become weak. They'll lose the ability to walk, talk, breathe, eat, use the bathroom, everything. Your muscles help you to move. Your diaphragm helps you to breathe. Your legs help you to walk. But if you lose the ability to move those muscles because those muscles are breaking down, the disease will lead to premature or early death. Okay? So let's practice building a pedigree of our own based off of a case study. So the condition we're going to be talking about is albinism. We had a wonderful presentation on albinism 2 weeks ago. And the inheritance pattern is that it's an autosomal recessive allele. So it's caused by a gene on chromosomes 1 through 22. It never, it equally affects males and females. There we go. And it is caused by a recessive allele. So it can skip generations. It's caused by a lowercase letter. So we'll use the letter A to represent the expression of albinism because it's a lowercase letter, lowercase a. So our case study, 2 normally pigmented parents have 3 children. The first child is a girl and their second child is a boy. They have normal pigmentation. Their third child, a girl, has albinism. And that girl marries a normally pigmented male and they have 4 children. The first 3, 2 girls and a boy, have normal pigmentation, but their 4th child, a girl, has albinism like her mother. Let's draw the pedigree. So the first thing we need to start with is 2 normally pigmented parents. So how do we denote the parents? Males are what? Squares. Oh, where's my annotation? Here we go. Males are squares and females are circles. So they're normally pigmented. They're unaffected. So A, if this is a recessive, undo. A is normal and lowercase a is albino. Okay. So 2 normal parents. They have 3 children. So let's write 3 children down here. So the first child is a girl. The second child is a boy. And the third child is a girl. But it says she has albinism. So what do we have to do? We have to shade it in. We have to shade it in. It's not going to be neat. Don't judge me. Okay. That girl with the albinism, she marries a normally pigmented male. So she marries a normally pigmented male. So he's not shaded in. And they have 4 children. So let's write 4 children. All right. The first 3, 2 girls and a boy, 2 girls and a boy, have normal pigmentation, but their 4th child, a girl, has albinism just like her mother. And if she has it, she's shaded in. Shaded in. Okay. So we've drawn our pedigree. This is what our pedigree should look like. Now we have to label the genotypes of as many individuals in the pedigree as possible. Shade in half of the symbol if you know that the individual is heterozygous or a carrier. So for ours, we are going to shade in halfway. Remember, not all pedigrees use the half shading, but for ours, we're going to use half shading. Okay? So let's label the genotypes of as many individuals in the pedigree, which means we kind of have to work backwards, right? So let's start with what we know, right? We know that albinism is a recessive, autosomal recessively inherited trait. So if you have 2 recessive alleles for it, you'll be albino. So, and that means you're shaded in. So we know the genotypes of these 2 girls are lowercase a and lowercase a. Okay. So, in order for this daughter in the last generation to be albino, she had to get 1 allele from her mom, but guess what? She had to get 1 allele from her dad. So her dad is heterozygous for the trait, which means he needs to be shaded in halfway. Sorry, y'all, shading is not the best. Okay. And so if we were to do a Punnett square, of dad and mom, the only 2 possible genotypes that we get are heterozygous genotypes and homozygous recessive genotypes. And if the student or if the child is homozygous recessive, it will be shaded in. They would have albinism. But look, their kids don't have albinism, which means what? Their kids are also carriers, heterozygous. So that means that their kids, don't laugh at my drawings, you guys, would also be carriers. Okay. Now, let's work our way back up. We know the genotypes of their kids. Let's go back up and figure out what her parents and her siblings' genotypes are. And this is something I forgot to do, you guys, and I'm so sorry. Let's label our generations. It'll make it easier if we label our generations. So this is generation 1, this is generation 2, and this is generation 3. Okay, and then we can write our numbers. 1, 2, 1, 2. 3, 4, 1, 2, 3, 4. Okay. So let's try to figure out generation 1's genotypes and generation 2's individual 1 and individual 2's genotypes too. So let's try to figure out this woman's siblings and parents' genotypes. All right, so we know that she's affected. She's got 2 recessive alleles to make her albino. And remember, she has to get 1 from her mom and 1 from her dad. So if her mom and her dad passed on recessive alleles to her, but they're not affected, that must mean they're what? Carriers. That must mean that they're carriers. Because there's no way she could have albinism if she didn't get 1 recessive allele from her mom and 1 recessive allele from her dad. She couldn't get both of them for her mom, and she couldn't get both of them from her dad. She had to get 1 from each in order to be albino. So both of her parents were carriers. Now let's look at her siblings. Sibling 1 and sibling 2 in generation 2. I'll move these out of the way so we can see. That's 1 and that's 2. Okay. So in order to figure out the possible genotypes, what will we have to do? Just like we did with our other parents, a Punnett square. So let's do a Punnett square so we can figure out possible genotypes of sibling 1 and sibling 2. So mom was heterozygous, dad was heterozygous, capital A, lowercase a, capital A, lowercase a, lowercase a, lowercase a. So look at this. We know our siblings are not affected, so they have the possibility of either being heterozygotes or homozygous dominant, not having the trait at all. But since we don't know what allele each parent actually gave for definitely to each of these offspring, we can just say capital A and leave a blank. Capital A, leave a blank. We know they're not affected. That's why we had that capital A. But we don't know if they're carriers or whether they're completely unaffected at all. We wouldn't know that unless we knew what their kids were. So leave the capital A blank, capital A blank. All right. So here are some summary questions to think about. What is this pedigree used for? What is a pedigree used for? How can you tell if a trait is autosomal or sex-linked? How can you tell if the trait is dominant or recessive? And then review one of the genetic disorders discussed in this presentation. I would rather you review the albinism case study and see if you can try to rebuild that pedigree on your own. That concludes my lesson for today. You guys have a wonderful day. Make sure you review, and I'll see you tomorrow.